Hi everyone, this is Nicole Wood, Wildlife Bio Gal, live from New Jersey. So today we're going to be doing a whole series of broadcast here at the Rutgers, New Jersey Institute of Technology combined campus. I guess that's probably the best way to say it. So we're technically at the New Jersey Institute of Technology, but this is also a Rutgers thing. So if you want to be able to follow along, follow the Scicomm Monday Twitter handle, and we'll be posting the broadcast up there. Also feel free to follow along with the hashtag, so everything that we're doing for the Scicomm trip here will be the Rutgers NJIT Scicomm hashtag. And then if you have any questions, also feel free to tweet me at my handle. So like I said, we're going to be talking to a whole bunch of different researchers today, so feel free to send in any questions. If we don't get to your question, if you're watching this on a replay later, feel free to tweet in your question and I'll get those passed on to everyone. So with that, I'm going to introduce our first guest, and why don't you have at it? Say hi to the world.
Hi everybody, I am Manpreet Kohli. I'm a graduate student here at the Rutgers side, but you know, as Nicole said, it's a mix. So we hang out often at the New Jersey Institute of Technology. I came to United States in 2010 after completing my bachelor's. Then I completed my master's in science degree here at Rutgers, and that's when I decided I think I wanted to do my PhD as well. So
you're like, you're here, you might as well just stick around, right?
I know, it is so much fun to be in academia. I've enjoyed my time here a lot, and I enjoy my research a lot. My advisor is great, so
that always helps. Having a good advisor is one of the most amazing things that helps you to stay in academia and go through stuff.
Yes, it is very important because you need their constant support, right? So I definitely got that at all points in my career. So yeah, so here I am about to finish, hopefully soon by next year. Yeah, that's me.
So for everyone out there, if you have any questions for Manpreet, feel free to go ahead and ask them away. We're going to make this kind of more of an AMA format, so send in your questions and we'll be more than willing to catch them. If we don't catch your question, make sure that you keep sending them in. We'll eventually hopefully get to it. So we'll be around here for about another 20 minutes to a half hour before I find out I'm getting handled and off to the next thing. So, but so yeah, why don't you tell us, like, what was your experiences, you know, coming from a different country to the U.S., being, you know, an international student? Like, how does that process work for those people who might be interested?
So I've kind of seen my experience in like two different ways. If I just think about it culturally, because I've always grown up in the cities, I did not find a lot of difference just adjusting to the culture here and, you know, just getting used to this big city here because, well, as I said, I've always lived in the cities, but academically it was a little challenging. My first 2 years when I was pursuing my master's and when I started doing my first project, it was a little challenging because we're not quite trained in the same way to do research in India as, you know, people are expected to to pursue their research here. You're expected to be quite independent, which I hadn't experienced before, and a lot of times students don't get to experience that because they usually just come on and they work with their advisor's project, so the advisor is kind of, you know, telling them how to, or what is their next step or what to do next, right? But with my advisor, it was free reign. You could do whatever you want, right? And she did not ever tell me, you know, you must do this or you should work on this project for me, right? So it was very free reign, but then that was all that it required me to be more independent than I was used to, right? And used to thinking, coming up with my questions independently and approaching them in my own way. Like I always thought, no, I should go and talk to somebody before I am doing this because what if I'm making some mistakes? So there was always that.
So you're saying in India it was more controlled with what you're doing versus here it's more free form?
Yes.
All right. Yeah, that's interesting because, like, I wonder if that's, you know, maybe a variation between different advisors and labs or maybe universities, because I know some professors are pretty, like, regimented with how they deal with their students here, but others are like the kind of laissez-faire attitude of where you're like, yeah, just go do science and you'll get something at some point in time. So I wonder if it's more like an individual cultural thing between like different mentors here versus maybe, you know, a whole, like, is the U.S. more relaxed than maybe what India is? It'd be interesting to like kind of tease apart some of those differences.
Well, you're right in saying that some advisors even here are more regimented than you know, others. So that's just like how they operate their lab or how they've trained, you know. So there's definitely that. But I think we were slightly more, I'm not saying to, I don't, this is not what I want the users to understand, that, you know, we were in a strict regime or something, right? It was not something like that. It's just, the independence was slightly less than it is here. Just that. Now, it could, of course, it can be influenced by the fact with people I worked with rather than other labs, because I've not experienced a lot of labs. So, you know, my experience in India, working in India, is rather myopic, right? Because I just worked with one or two people, right? And then I was working in an pharmaceutical industry for a while, so of course there you have to follow what they're saying, right? So there's definitely that, but I do feel it might be slightly more
still more free-form here over here.
Right, yes.
Yeah. Well, we have someone, it looks like maybe that was shout out from France. So hello, I'm assuming if you say you're French that you're from France. I could be totally assuming things, but if you are, great to have you tuning in here. So if you have any questions, make sure you send them in. We're talking with Manpreet about her research here at Rutgers University, even though we're technically on the New Jersey of Institutes campus. They are kind of a combined program here when it comes to biology. So any questions you have about that, feel free to send them in. So why don't you talk to us a little bit about your research, because I see some really cool things over that I'm super excited, because, you know, I just watched Jurassic Park a couple of weeks ago, and I'm just really kind of like geeking out about what you got over there. So why don't you tell us about your research?
So as I was talking to you before, I am interested in macro and micro evolutionary processes that sort of guide the biodiversity that we see around us today and what led to this biodiversity, right? And events of the past, like mass extinctions, especially the one that took dinosaurs also, including that one, have, influenced the kind of biodiversity we see today. And one of the things that I am working on in my, for my dissertation is sort of using the information that you get from molecules, that is DNA, right, and combining that with fossils, right? So what I have here is an amber piece, right? So it's a little amber with a dragonfly wing in it, which is probably hard to see on the camera, but
it looks really cool in person, I will tell you that.
So what I would do with this is, sit under a microscope and make drawings of the features that I can see in the amber, and then take those drawings, and if there were different bodied parts, I would try and combine them together to sort of make the complete dragonfly. And then look at the features, look at the morphology, and then try and identify which group it belongs to, right? You would try and find out which family or genus or species, if possible, to try and identify it to, that level. And then a lot of time, paleontologists and molecular biologists, right, they would use these fossils for what we call calibrating our molecular clocks. And very briefly, molecular clock is the idea that, your genes are ticking or mutating at a specific rate, right? So say it changes one nucleotide 1 million year, right, for every million years. So if I knew there were 30 differences between, two species, then I would say, well, they became different 30 million years ago, right? But then you kind of need to calibrate your clocks, you need to tweak it a bit, and that's where fossils come in. Like, you can use fossils like this to sort of inform, okay, I saw this kind of dragonfly, say, 5 million years ago, so I can give the model that information, and then you would, do some more statistics on it to figure out, like, how old two species are. So that is some of the work that I do here as a graduate student,
So, like, how common is it to find fossils like that? I mean, when you watch Jurassic Park, they make it seem like, oh, yeah, they're finding those mosquitoes and getting the blood out of it, like, every 2 days or so to be able to get all that diversity of the species that they have there at the park. In reality, like, how rare is something like this to be able to find like an actual insect in amber from 5 million years ago? I mean,
so just starting from the Jurassic Park point of view, it is partially true in the sense that it is easy to find insects trapped in amber, right? You would find a lot and lots of insects, like especially ants are very abundant. Flies, which belong to Diptera, are very abundant in the amber fossil record, right? Because they are easy to get trapped.
Oh, that makes
versus
sense.
Like
something that's
gigantic, like a lizard. You would almost never find
Right.
like a lizard in an amber piece, right? So these insects get easily trapped, right? So it's easy to find these insects in amber pieces. However, extracting the DNA from it and then doing all the magic done in Jurassic Park, that is a big stretch, because you
kind of more that science fiction side rather than the pure science side of it, maybe.
Yeah, it's just because it's impossible to get anything out of those insects that are trapped in there, because if you think about it, it's just the shell that is left, but nothing's really left inside, because that's been just sort of replaced. So you can see it's just a shell that is left. You could if you do CT scans, which a lot of my friends do who work with fossils, you can see internal structures that sort of become hardened, right? And you can see internal structures. But extracting DNA is not that stable that it will last for millions of years, right? You could potentially get some protein, but that's like a really, really advanced area of research which people have just sort of started to, work more on. So yeah, a bit of a stretch about the extracting the DNA and bringing those giant beasts back to life, but definitely it's easy to find a lot of insects in amber. You can find it on eBay. People sell a lot of jewelry with just like insects in it. So yeah.
So, like, regionally, like, where would you find them? Like, I know 5 million years ago, a lot of places, like, say, where I was underwater.
Right.
So, like, where would you, like, find places where there would have been insects? Like, is it, like, are you finding them around here? Like, where would you be finding your fossils mostly?
So, we do have fossil deposits in New Jersey.
Okay.
But, I want to say they're not amber deposits, right? But there are certain regions which are very common for finding amber deposits, like Dominican Republic, or like, for example, this amber piece, this comes from the Baltic region. So those fossil deposits have yielded a lot of amber, like tons and tons of amber that you can find, which then you sort of sift through and you look for what you're looking for. So it's common in those areas. I wish I could speak about New Jersey, but see, this is it. I'm not a paleontologist, so I don't know that part.
So if you're a paleontologist out there and you know anything about New Jersey fossils, let us know. We'd love to hear about it for sure.
Yeah, absolutely.
So another thing too, like you were talking about before we went live on air, was where you actually find dragonflies, and you were saying something about the Arctic with dragonflies, which was blowing my mind. So can you expand on that a little bit more?
Yeah, absolutely. So usually when people think of dragonflies and damselflies, you're like, we go out on a pond on a sunny day, and you will see them. Yeah, they'll be there flying by the edges of the ponds, which is where you would mostly find them. But then as you move further north, and this is not just with dragonflies, I'm sure you see this in all organisms, right? The further you move away from tropics, or the closer you go to the poles, the diversity decreases, and the basic or the most important reason is that it is because of colder environment, right? So people don't think of insects in general living in colder environments, right? But there are, of course, some species which are doing very well, who are adapted to live in these colder environments. And one of such species of dragonfly is what I'm studying, and it is called the northernmost dragonfly, because I think the definition for that is that there is no other breeding dragonfly, which is found as north, right?
Oh, okay.
So some of the populations of the species, Somatochlorus salbergi, I'm gonna just plug that in.
Throw that in. I mean, you might as well, right?
Might as well, right? Is found as north as 72° north, which is much further from Arctic Circle, right?
Yeah. So for those of you, for just comparison, I know the 45th parallel goes right through the Lower Peninsula of Michigan. So think of how much further north that is. So yeah, that's pretty impressive.
Yes, it is. So that's what I've been trying to investigate. Like, wow, how is this dragonfly species like surviving this cold environment? Because clearly most of that region is frozen for most part of the year, right? So the dragonfly larvae, which are the nymphs or the babies, which are living in the water, right, must be surviving all of that cold. So I'm kind of trying to see how they're doing that. And plus, the most fascinating thing is that this dragonfly species is found all around the Arctic Circle.
Oh, really? So it's not just like regionally, so like not just in North America, you're finding it over like in Asia and Europe.
Yeah,
oh, that's cool.
So all across the Arctic Circle and above it, this species is found in Alaska, in Yukon, which is in Canada, right? And then you find it at the tip of the Scandinavian Peninsula, which is the Norway, Sweden, and Finland, and then northern parts of Russia, and then we suspect there might be some on like the eastern side, or the eastern side on the Japanese side also, but we don't know that yet.
Ooh. So that should be a fun thing to find out.
I know, but so it's crazy
Field
that
trip.
it goes. Yes. Oh yeah, that's definitely true. I traveled to Yukon in 2015 to do this incredible field trip to collect these samples, and I must say it was one of the best field trips I've ever taken. It was incredibly beautiful. We were out in the wilderness without
middle of
nowhere.
How bad were the bugs out there? Like, were you having to like full on have the nets over everything?
Yeah, we did, because actually we got lucky for most part of our collecting trip that we didn't encounter those black flies. But there were days where you were like, I cannot even open my eyes without having a net on my face, because it's
that would have
really
made like
bad.
a great if I don't know how many people follow along on Twitter with the actual living scientist thing, but that would have been a perfect picture for that to like showcase.
I might have a picture somewhere, but
if you do, send it and I'll retweet it out so everyone can see it, because that would be amazing to see, like from live from the field. Kind of pictures like that are always great.
Absolutely, I will do that. But yeah, that was a very fun trip, and as you said, it was in the middle of nowhere. It was funny when we were just like, so there's this town in Whitehorse, that's the in Yukon. There's the last place you can fly into, and after that you have to do everything on road, right? And about, I would say, 70 to 80 kilometers out of Whitehorse, it said, you are now leaving the 911 region. So basically you have nothing. You have nothing. They're like, you're on your own now, not our problem. So that was quite interesting. But so this dragonfly species is everywhere, and that's what I'm trying to find out. How is it possible that you have populations in Russia and in Scandinavian Peninsula, but then you also have populations in Canada also? And this is the most interesting part I've found, which I should publish soon, is that I see no genetic differences.
Yeah, that's right. Like, you would think there would be like either subspecies or something, like you could start seeing that drift of them apart.
Exactly. But, I started my research thinking that. I'm like, surely, because there are populations here in the Scandinavian Peninsula and in like Yukon, you've got to be different from each other, right? And then I sequenced DNA, which is what I do on a daily basis in my lab, and I sequenced some DNA.
That's my like regular time.
But and we found that these populations genetically are absolutely, I don't want to use the word identical, but they are almost so similar to each other, which is surprising. That how are they so similar, even though they are apart, thousands and thousands of miles, right? I mean, there's Greenland and the Atlantic Ocean in between, and how is it still similar? So,
do you think like there might actually be some migration, that they're being able to keep that gene flow going between those populations, so they're staying similar, or do you think that they just aren't diverging at all?
So those are excellent questions, and that is exactly what my research will focus on in the next, coming years. I literally wrote a grant yesterday to ask them money for,
Fingers
studying.
crossed that it actually gets funded.
Yeah, to ask for some kind of money to, do migration studies and see if they are indeed migrating, if I can, pick up some kind of isotope signature to maybe see if there's a possibility that they are migrating. Otherwise, my guess would be that they've not had enough time to, just diverge, and it's very recent populations which were formed right after the ice age, the recent ice age we had, which was it finished around, 10,000 to 12,000 years ago. So maybe they just, became separate then. Right. So that's why I don't see any signature, in their genes. But very fascinating.
Or we need the engineers to actually create the little tiny backpack so you can track them. I mean, wouldn't that be amazing?
So much fun.
Put a backpack on a dragonfly and see where it goes.
We need that. I mean, people have tried to do that before, like put some radio transmitters on them, but they are really big. They're still the size of those radio transmitters are big for dragonflies, and they put it on
with birds. Like sometimes they're still like too huge for some of the bird species we have, and so like I can only imagine a dragonfly. I mean, while there are some decent sized dragonflies, like I was surprised like they're actually big enough that they catch butterflies. Like I saw one grab one out of the air once. I'm like, holy crap, that's that big, and that to do that. So I mean, they do get decent size, but I mean, you're still talking about an insect and like that weight on it, so it can still fly and do what it naturally should be able to do.
Exactly. Like, so I mean, you don't want to, affect your experiment because the dragonfly were not in their natural state, and it'll affect your results. So they've done it on bigger dragonflies, which are about yay big. I guess that's not very good here, but they're like yay big. But my dragonfly species like this small, so they're not big enough to carry those. So we definitely need yet smaller, trackers that can work on them across continents. Also, that's a big range.
Yes, we need the GPS thing on there. Yeah, because especially if that's the question you want to answer is like, how are they globally? You need the GPS. You can't just like be out there radio transmitting as much.
Exactly.
Like as far as like seeing where they like, do you try to do any tagging? Like do you like put like color tags on them? So like if you like catch them again, like you see, oh, we tag this one here, and we're seeing like 50 miles away. Oh, we're finding them this far away. Is like any of that been done with them?
So no, I haven't done that with my species because they are kind of elusive, right? So and also they are found in these environments which are like really hard to get to. I mean,
so like the amount of time you have to sample there is really tough.
People do mark and recapture studies with insects, with damselflies. One of my colleagues, who's also just recently finished her PhD here, was doing that with damselflies in the tropic, where she would, put numbers on their wings, and then she would try and collect those individuals again, like next day or after a week or 2 weeks, just to see how long they live or what is the population size. You can definitely do that, but I haven't been able to do that with my study system.
Yeah, it's one of those things, like it would be like if we had all the time and money where you could just like be out there in the field to like really do all that, but like you're so limited with the time, especially when you're in the middle of nowhere. I mean, just the funding to be able to support that, to get out there. I mean, you're talking your plane ride, and then I mean, if you're in the middle of nowhere, you probably had to have someone with you to make sure that the bears weren't attacking you and things like that. And so like all of that like costs a lot of money, and to be able to fund those studies, like it really is like limits what you can do out there.
It does. I mean, I do need a lot of funding just to like it costed me a lot of money just to get to Yukon and plan our whole trip, because it's a very rugged domain, and it's not, so you've got to rent a car, and they won't just give you like a small car because they're like, oh, it won't go on that kind of road, so you need a big car, and and then you need a lot of equipment, so it kind of does become financially challenging to keep doing your project if you don't have a lot of funding.
That's one of the things I think a lot of people forget when it comes to science, is how much that funding is important to be able to help answer these questions. And some folks out there might think, well, what do I care about a dragonfly? But the questions that you can answer with that and how that impacts that greater ecosystem is so important. We need to have those answers, because we see what happens when we don't have the answers. All of a sudden the ecosystem just goes haywire, and we're like, we don't know what to do now. But if you had that knowledge and that data set, you could be like, okay, this is how we can like fix this and get back to that normal state.
It's definitely true. I mean, so I have a little example just to sort of speak to that, because not a lot of people have worked on this species, but some people have dedicatedly worked on it for a long time, right? So there are these twin brothers who've been working on it, for a very long time, and they made their records in 1980s. Around most of the records for this species in Yukon come from all the work that they did during 1980s, and I followed their exact chart where exactly they said this is where I found the species, this was the puddle where I found it, and I tried to go back and sample those exact sites, and believe it or not, I did not find the species till the last day of my trip, because most of the sites where they had recorded, most of the these ponds where they had recorded these species had disappeared, because the permafrost beneath these ponds, because we are above the Arctic Circle, so we are in the kind of the permafrost region, right? And as the permafrost collapses because of increasing temperature, the whole pool collapses. So that's why these species have lost their habitat, right? And people can argue, all right, it's one dragonfly species that's gone, but the thing is that it's not just that dragonfly species, and it's not just my species that disappeared with the disappearing of that habitat. It's a lot of other insects and other animals that depend on that, because there are insects and there are birds that depend on that insect, and that there's a lot of other organisms that depend on those birds, right? So it's affecting the whole ecosystem, and that is just kind of crazy to just see that. They're like, the ponds are gone, there's nothing here, it's just like
as soon as like that one food source goes, it like just goes right up on the system. And it's like next thing you know, a dragonfly could be in theory impacting the polar bear that's there. If you don't have that dragonfly, it goes up to the point of you might not have polar bears because of that cascading impact.
It could, not so directly, but it probably would. I agree. So, it is incredible.
So we're gonna start closing up here. So if you have like any last questions, get them in right now. Otherwise, feel free to tweet me afterwards. Follow the Scicomm Monday hashtag, the Scicomm Monday account. That's where we'll be posting all of the broadcasts coming throughout the day. Follow the Rutgers NJIT Scicomm hashtag for everything that's going on. And then if you have questions afterwards, either tweet me at Scicomm Monday or at my personal handle of wildlifebiogal.com. Just a plug for the next broadcast at 10:30, I believe it is 10:30. Yes, I think that's what it says. 10:30 is with Tony Cullen, and we're going to be talking about his work here at Rutgers. So with that, I hope you guys all enjoyed it. Make sure you keep tuning in, because there'll be a whole bunch of broadcasts throughout the day with all the amazing researchers here in New Jersey. So thank you very much. It was very much enjoyable to have you here. And let us know about any cool amber stuff, because I'm still kind of geeking out on it. So as soon as we close this off, I'm going to be looking at that a lot closer, because it's really neat. So I hope you guys enjoyed this. I hope you had fun.
Yes, thank you so much for having me here. This was a lot of fun. Thank you.
Great. So like I said, keep tuning in, and we'll be posting up these broadcasts throughout the day.